work, unfortunately, at the moment.
So, good afternoon, everyone. My goal today will be to tell you about the effects of sex steroids on cognition in aged non-human primates, and in particular rhesus monkeys and marmosets. So this is the outline of my talk. First, I'll briefly describe human cognitive aging. Then I'll talk about studies that have investigated the effect of estrogens on the brain and cognition in aged female macaques. Then I'll turn to males and we'll describe some of the studies that we did in male macaques. And at the end, I'll tell you about recent studies that we did using a different model, the marmoset. So first, I would like to talk a little bit about human aging. And so this map shows you the percentage of population over 65 years old in the world in 2008, with the dark green areas representing countries with more than 11% of older people. So you can see that a lot of countries are affected. Japan was leading the way, but more alarming are the projections for 2040. So you can see now that almost all continents except Africa are affected, have a lot of older people, and so this illustrates the fact that our population is aging, and is aging very rapidly. And this is concerning if we think about cognitive aging. So these are data from Dennis Park and collaborators showing you age-related cognitive decline in different cognitive domains in humans. So as they age from the 20s to the 80s. So you can see there is a linear decline with age, with only one domain being spared by aging. This is verbal knowledge, so vocabulary actually increases with age, but otherwise all of us will follow this trajectory, and obviously it's important to try to find interventions to alleviate cognitive aging. So the intervention I'm going to talk about is hormonal therapy, and so why would hormones have anything to do with brain function? Well, as you know, in women there is a dramatic loss of estrogens at menopause, so around 51 years of age. In men, the decline in testosterone is much more progressive, but is significant also. And so the question is to know whether the loss of sex hormones has anything to do with memory loss, and if so, can we just replace people with sex hormones to alleviate age-related cognitive decline? So why would estrogens affect brain function? There are a lot of reasons for that, and this is just a summary of the effects of estrogens in the brain. And here, that comes from an excellent review from Roberta Brinton and collaborators that shows estrogen receptors in many cognitive areas of the brain, and especially in the prefrontal cortex and hippocampus, which are the two areas that I'm going to focus on today. So in both these areas, estrogens increase the number of dendritic spines. And this is important for learning and memory, because this is the spines are the sites of synaptic contact. It enhances also long-term potentiation in the hippocampus, increases neurogenesis in the dentate gyrus, modulates all neurotransmitter systems, and estrogens have a number of neurotrophic and neuroprotective properties, which could all explain effects on learning and memory. So here I focus just on the effects of estrogens on the dendritic spines, with an example here in the rat. This is a pyramidal neuron, and you see here a dendrite with a little protrusions are the dendritic spines. And here you have a female rat that has been ovarectomized, and which is not replaced with ovarian hormones, compared to a female rat that is ovarectomized and replaced with ovarian hormones. You can see there is a dramatic increase in the number of dendritic spines. Here's just a graph to show you that this phenomenon also occurs in the prefrontal cortex and also occurs in primates. So, knowing this, what is the evidence that estrogens actually benefit cognitive function in women? Well, in women, the findings are actually very mixed. And here I need to take some time to talk about the largest trial ever conducted on the effect of estrogens on on health outcome in women. That was called the Women's Health Initiative. That was a huge study conducted on more than 27,000 women across 40 US centers. And as part of this huge study, there was a Women's Health Initiative and memory studies that focused on cognition. So for this trial, women without a uterus were randomized to estrogen alone or placebo, estrogen in the form of conjugated equinestrogen, and women with an intact uterus had, in addition, a progestin, medroxyprogesterone acetate. I need to point your attention to the fact that these women were very old. I mean by that they were 10 years past, more than 10 years past menopause, which is important to explain the results. The duration of treatment was on average 5 to 7 years because, as you probably know, the trial was terminated early because of negative effects on a number of health outcomes, including breast cancer, blood clots, and so on. For cognition, everybody expected beneficial effects of estrogens based on preclinical studies and on small trials with women. However, exactly the opposite happened. So this is an estrogen plus progestin group. For global cognition, there was actually a significant impairment in the treatment group compared to the placebo group. And for dementia, it was not better. In fact, after 1 year of randomization, the treatment group had twice the risk of getting dementia compared to the placebo group. So, as I said, these results were completely unexpected and really contrast with the results of rodent studies, which consistently find beneficial effects of estrogens on the brain and cognition. And so, I think it's very important to have primate models to try to reconcile these findings, and especially to determine whether the mechanisms that are identified in rodents also apply to primates, to predict the therapeutic effects of treatments in humans, and to design new therapies that would be optimal for human cognitive health. So the primate model of choice for these studies has been the macaque. So Macaca mulata, it's a rhesus monkey, or fascicularis, it's a cynomolgus monkey. They're interesting because they age 3 times faster than humans. They have a relative long life, about 30 years old. They have a complex brain, and their patterns of cognitive aging are very similar to ours. With regard to endocrinology, they are very also similar to us, both for the male and the female. However, with regard to menopause, there is a big difference. The female rhesus monkey gets menopausal very late in its lifetime, so basically a few years before they die. So it's very important to understand that all the hormonal therapy studies that have been conducted in monkeys so far have been done in ovarectomized monkeys and not naturally menopausal monkeys, because these monkeys are just too old, first of all, to find and to test for cognition. So in one of these studies by Peter Rapp and collaborators, they tested older over-ectomized monkeys, rhesus monkeys, on a battery of tasks, including the delayed response task, which is a task of working memory dependent on the prefrontal cortex. So just to show you how it's done, here you just show to the monkey where the treat is going to go, either on the left side or right side of the board. The two sides are covered with identical stimuli. There is a delay. The monkey has to remember where she saw the treat, and here she makes a mistake. But she just has to keep in mind where the treat was. Okay, and you can see here that performance decreases as a function of delay. This is a classic result. But what was really amazing is that with estrogen replacement, estradiol, CQNA, the monkeys were much better at every delay on this task. We also tested older over-ectomized monkeys in a battery of tasks, including the task I just showed you. But these monkeys were over-ectomized for an average of 12 years, so it was really long-term estrogen deficiency. And interestingly, we didn't find a beneficial effect of estrogen on that task when we tested these monkeys, which is consistent with that idea that you must give estrogen at the time of estrogen loss to have beneficial effects on cognitive function. However, in another task, the task of spatial memory that I'm going to briefly explain, we found a beneficial effect of estradiol treatment in these monkeys. So here it's a task where the monkey is presented with identical stimuli, this brown disc on a board, and they have to choose the disc that is placed in a new location at each trial. Okay, so here she just saw this one, so now she has to go for this one to find the tree. What is interesting in this task is that with each correct answer, a new disk is placed on the board, so it increases memory load. So it's really a task of spatial working memory. In any case, so when we gave estrogen to these monkeys that were over-ectomized for a long time, there was a beneficial effect of estrogen treatment on this task. And these graphs are from the work of Peter Rapp and John Morrison and collaborators, where they looked at the number of dendritic spines in the hippocampus and prefrontal cortex. So the two areas that support performance on the task that I've just shown you. And you can see that in both young and older monkeys that are over-ectomized, there is an increase in dendritic spine with estrogen treatment. Okay? That occurs for both the prefrontal cortex and the hippocampus. What is very interesting is that you can see that the older monkeys in the prefrontal cortex have a lower number of dendritic spines than the younger monkeys, right? And estrogen is able to restore the number of dendritic spines to the level of the young over-ectomized monkeys. So there is a partial restoration of dendritic spines to the young levels, if you will. So to summarize on this part, on what we know about estrogen and older female macaques, we know that estrogen is able to enhance selective aspects of cognitive function, both for tasks that depend on the prefrontal cortex and the hippocampus. These cognitive benefits may be mediated through this increase in dendritic spines. This is the hypothesis that is out there. It's very difficult to demonstrate that it's actually the mechanism. But because these spines are lost with age and can be partially restored with estrogen, it's really, if it's a correct hypothesis, then we have here a potential target to reverse the cognitive, the detrimental effect of aging on cognition. So now I'm going to turn to males, and So this is quite incredible. In this field, males have been neglected. In fact, until we started this work, there was absolutely nothing on the effects of estrogen on cognition in male monkeys. As I told you earlier, so there is a gradual decline in testosterone with age in humans. This decline starts early, but is very progressive, so it's about 2% per decade. You can see here. This decline is associated with a number of detrimental effects, so loss of bone, muscle mass, increase in fat mass, impairment in physical and sexual function. It is still not clear if it is also associated with an impairment in cognition. Testosterone, just like estrogens, have a number of effects in the brain. There are androgen receptors in brain areas that are important for cognition and emotion. Testosterone increases also dendritic spine density in the prefrontal cortex and the hippocampus. That has been demonstrated in rodents, not in primates. And they also have a myriad of effects in the brain, neuroprotective and neurotrophic effects. So the evidence that testosterone actually benefits cognition in older men is also very mixed. If you look at endogenous testosterone and cognitive function, some studies find an association between higher levels and better performance, but many studies find absolutely no association. If you look at studies that have investigated testosterone administration, again, you have some studies that find beneficial effects and others that find no effect or negative effects. When we started this work, there were very few studies in male rodents and absolutely nothing in monkeys. So for this study, we decided to use a chemical castration paradigm. It means that the monkeys were intact, but we gave them a drug, lupron, which is a gonadotropin-releasing hormone agonist, to suppress their testosterone levels. It's a reversible procedure. So it was a crossover study where monkeys were tested at baseline without any drug treatment. Then they were treated with lupron, and 4 weeks later they have very, very low levels of testosterone. And after that they were treated with testosterone or oil in a crossover design. And during that time we tested them on a battery of cognitive, motor, and emotional tasks. And we had 16 monkeys, which is not a bad sample for a cognitive study in monkeys. We had 10 old and 6 young monkeys. I'm just going to show you the cognitive task results for the older monkeys, briefly describing the task that we gave them. The delayed non-matching to sample is a task that depends on the medial temporal lobe, where they are shown a sample object, and after a certain delay, they are shown the same object and a new one, and they have to select the new one to find the reward. So first they have to learn the rule, and then they're presented with different pairs of objects like this. So these are the results for the older monkeys, but the results were identical for the younger monkeys. There was an effect of delay that was expected, but absolutely no effect of treatment. We also tested them on the DRST, so this is the task with the little brown discs that I showed you earlier, a task dependent on the hippocampus. Here we look at different measures that I don't have time to detail, but there was also absolutely no effect of treatment on that task. And we tested them on the delayed response task, where they just have to remember where the treat went, left or right. And here too, there was absolutely no effect of treatment. We also tested them for fine motor ability, and it's a task in which we just look at the time it takes the monkey to remove lifesaver candy from shapes of different complexity. This is a task that is easy to give and that the monkeys love to do. So, and it's very sensitive to age-related decline. And we were hoping to treatment, but again, so here you have the young monkeys and the older monkeys, and there was no effect of treatment for any shapes. In that particular study, we also looked at emotional reactivity, because I was suspecting that testosterone would actually affect this domain. And in one aspect of the experiment, we gave monkeys some videos to watch, videos of unfamiliar monkeys engaged in different activities, fighting, so that was a negative video, grooming, a positive video, and sleeping, so a neutral video. Unfortunately, I think that some of the videos do not work in the computer for some reason, so I can't show you that. But oh, now it works. So that's just a grooming scene, so very relaxing, and that was a fighting scene. I don't think it works here. Anyway, so both the young and older monkeys preferred watching the negative scenes. So it may not be a surprise for anybody that they were more interested in looking at this. What was more interesting is that under testosterone, they were watching these negative scenes significantly more. So it seems that testosterone increases attention to social threat. At least that was our interpretation at the time. We also gave the monkeys a task of anxiety with approach avoidance. So for that task, the monkey is placed in a cage with 2 compartments, and there is a divider here, and they don't know what's going to be on the other side. It could be a neutral object, a novel object, or an aversive object like the Shrek mask, which actually is very aversive for the monkeys. And here there is half a banana to facilitate approach of the object. What I've shown here is just the amount of time, the proportion of time that the monkey spends in this compartment close to the object. And you can see that they spend less time when it's an aversive object. This is for the young monkeys and the old monkeys. For the young monkeys, there was no difference with testosterone treatment. For the older monkeys, the only effect that we found was that they spent more time in proximity of a novel object when they were on testosterone. So it seems that testosterone decreased their anxiety, but just for mildly stressful stimuli. It didn't work at all with the aversive stimuli. So to summarize on this study, it seems that testosterone manipulations do not affect cognitive function or fine motor function in young or older male rhesus monkeys. It's actually consistent with studies in men who use the same kind of chemical castration that we used with Lupron. And thinking about this, it may be that actually this treatment that we used has something to do with these negative effects, because Lupron not only suppresses testosterone, but also LH and FSH. And there are more and more studies that show that LH may actually be impairing cognitive function in primates and also rodents. So it may be that by lowering LH, we basically negate any effect of testosterone replacement. So this is something that we are interested in investigating further. And despite this, we had testosterone effects on some aspect of attention. It seems that testosterone modulates attention to social threats and may decrease anxiety in older monkeys only, and only for novel objects. So now that I've told you all about the macaques have also limitations that are sometimes difficult to overcome. They're large, they're costly, and they're difficult to maintain in captivity, and so you have to have studies with very small sample size, unless NIH suddenly increase their budget. They also carry a virus that is potentially lethal. There is poor availability of older individuals, and they have a long lifespan, so 30 years, that is not well suited for longitudinal studies. So recently, I've proposed to use a different model, the marmoset. The marmoset is more distantly related phylogenetically from humans and macaques, but they have really great advantages. The first one is that they're the size of a rat, and so they are very easy to handle. They are highly fertile, highly social. They do not have major biosafety concerns, and they are going to become the mice of the primate world, because transgenic marmosets have been made and are being made in these countries, and so this model is going to take off in the next years really dramatically. They've been proposed as a new model for human aging. One of their great features is that they're considered old by the age of 8. So we could really study them in theory for their entire lifespan. And with regard to brain aging, they show beta amyloid deposition, they show reduction of neurogenesis in the dentate gyrus, decrease in white and gray matter. So these are all features of human brain aging. And in terms of cognition, they can perform a variety of cognitive tasks, also touchscreen, and they can also be trained for conscious neuroimaging with minimal training, which is really great and different from macaques because of the size difference. So I'm briefly going to describe, for the few minutes I have left, a study that we did recently in the marmoset, looking at estrogen and cognition. We had 12 females that were ovarectomized. They were trained on a bunch of tasks. Then they were ovarectomized and replaced and treated with estrogen-filled capsules or blank capsules. And I'm only going to talk about 2 tasks, the object reversal task, which is a task of cognitive flexibility that depends on the prefrontal cortex. In that task, the monkey is presented with 2 objects, and one of them is always rewarded. And of course, the position of the object changes randomly. So you just have to figure out which object has a treat underneath. Once they know that perfectly, we change the rule on them. So now they have to pick the other object to be rewarded. And we give them 3 such reversals. And this is what you see here. So this is the initial discrimination, and then reversal 1, 2, 3. In red are the estrogen-treated animals, and in white the placebo-treated. In fact, we found detrimental effects of estrogen on this task. It was significant for the second reversal. And the animals also were making more and more perseverative errors in contrast to the placebo-treated animals. We also gave them the delayed response. I just have a little video where you can see a marmoset performing the task. You can see the size, they're really tiny. So here she saw the treat going, she needs to remember where it was. So first she goes to the wrong one. Here we go. So, oops. So in that task also, it was only a trend, but I don't know if you see it very well, but the estrogen-treated animals tended to perform more poorly than the placebo-treated animals. So that may be because we use high estrogens. This is our hypothesis, at least, that the dose that we use may have been too high for this test, dependent on the prefrontal cortex. And we're going to do now studies with a lower dose and also including tasks of that depend on the hippocampus. But despite this, I think we've shown that the marmoset can be a valid model to investigate the effects of hormonal therapy on cognitive function. And there are a number of questions that we want to investigate further. I didn't talk about young animals so much, but one of the consistent findings from this literature is that young monkeys, macaques, are pretty insensitive to hormonal treatment. Many groups have tried to manipulate estrogen levels in young monkeys, and it simply doesn't work. Cognition is unchanged. Whereas in older monkeys, you have clear effects of estrogen treatment. So, we're thinking that this is actually local estrogen produced in the brain that may be important. And it may be, and we do not know that there is a decline in local estrogen production in the older monkeys. Therefore, they would be more sensitive to estrogen circulating at the periphery, whereas young monkeys may have so much estrogen in their brains that it doesn't matter what you do at the periphery. So this is something that we're actually investigating using aromatase inhibitors in the marmoset. Another very important question is to find the optimal regimen of hormonal therapy. Till now, nobody knows what is the ideal formulation, the duration of estrogen therapy, the regimen that should be used. Some people have proposed that cyclic treatments are better. It's an interesting hypothesis, but it needs to be really tested. And I think the marmoset is ideal for that. We need to know more about androgens and cognition in males. I mean, the study I've shown you is actually the only one that is out there. And a lot of men are taking testosterone without knowing what it does to their health. So it's really an important subject. And finally, another study that we are trying to develop with a collaborator from the University of Maryland, Jessica Mang, is to look at the interaction between estrogen loss, sleep disruptions, and cognitive function. Because a lot of menopausal women not only have cognitive dysfunction, but also sleep problems, and it may be all related. So this is what we are trying to look at now. And with this, I thank you for your attention, and I want to thank my collaborators, students and postdoc, and of course funding, that is crucial. So, thank you. If you have any questions.
So you've got Craig Ferris in the mix. Have you got data on any MRI, functional MRI
I wish. So we actually scanned a bunch of monkeys, and so the procedure works. Unfortunately, the images that were obtained were not good enough for publication because there was a problem in his coil at the time. And we couldn't, that was made out of R21, we couldn't redo the experiment. So I can tell you that the procedure works, and that he actually did other studies in marmoset looking at the effects of ecstasy on brain function and obtained beautiful images. Unfortunately, for our study, it didn't work so well. But.
So I have another question. In many species, not only the primate, there is very poor literature on localization of androgen receptors, and in primates, in particular, the studies of where and when estrogen receptors are expressed is way behind some of the rodents.
Mm-hmm.
In that way, do you have any plans to look at ER alpha or AR in primates? Are there any antibodies that work?
So some people have done that. So there's a group in Oregon, Steve Kohama and so on. I've looked at estrogen, the localization of estrogen receptor, I think beta at least, in macaques. So there are people who are doing this kind of. I'm not doing this kind of work, but I think we know a little bit, even in humans, about the localization of these.
But usually they're limited to only, I mean, it's very, very rare to find one where they looked all over the whole brain or...
It's just because, coming up with enough sample, I mean, it's, these monkeys are very expensive and you don't want to, to sacrifice them to just, look at estrogen receptors or androgen receptors. I mean, but there are a few studies out there. So, Steve Kohaman, I think in the males, there is a paper by Abdel Gadir, and I can't remember the other. It's an older paper, but they had looked at androgen receptors. But again, it's limited with the subject, So it's difficult to come up with enough sample.
So have you looked in the overactimized females, speaking of Jessica, at things like temperature control in those animals? Do they have hot flashes?
This is what we're looking at, what we're trying to look at with quite some difficulty. So we don't know if they have hot flashes. That's we still we're looking at that. We actually have implanted two marmosets with a telemeter that records temperature, and but it's not hot flashes per se. So, but we see some changes in temperature under certain conditions that could be consistent with hot flashes. But we have not secured the money to go further than that. Let's put it that way. But this is something that it would be very interesting to see, because one of the hypotheses that we have is that actually estrogen loss probably induces hot flashes, and those which occur at night may be particularly disruptive to sleep, and then induce cognitive dysfunction that way. I mean, that's a possible route, at least, and this is what we are trying to disentangle.
So what we see in rodents that's quite profound is that if an animal has any interruption of sleep, then they end up with massive increases in histidine decarboxylase and histamine functions. So it could be, I know Jessica thinks it's all happening in the preoptic area, but it could really be there is a connection of histamine to that area as well, and it would be very interesting.
Well, it would be worth investigating, that's for sure. So we're trying to develop that aspect of the research and looking more at sleep and cognition. And in the marmoset, it's much easier to do terminal studies. I mean, just because you can have more animals, and anyways, they don't live that long either. So these type of studies are more feasible.
In this testosterone-treated animals, the males, you said that they watch more negative emotion video fighting. So in the clinical trial, testosterone, they have really shown men becoming aggressive. And some of the wrestlers, they confess that they become aggressive and they take the steroids. So no wonder that the monkeys are watching the fighting videos. Now, in another finding with those testosterone study, it improved the spatial cognition of the men. Like, for example, if you give them a map and ask them to go in a way they are able to coordinate and go to particular places. So have you carried out any study to see whether the testosterone improved the spatial cognition of the animal?
Well, we tried with that task of spatial memory, you know, that I showed you, you know, with a little disc, and testosterone didn't have any effect either in the young animals or the older animals, which was really disappointing because there is a sex difference in that task. Like males are better than the females, something that we've shown years ago. So I was really expecting testosterone to have an effect, but it seems that at least with the treatment we use, that's also the problem, is that this is with this particular treatment. You know, they were treated only for 1 month of testosterone. It may not be long enough. You know, the dose may not be appropriate. I mean, that's the problem. You may have to give, you know, testosterone for a longer period of time, or in a more cyclical manner. These are, you know, open questions.
So another question is amyloid protein. You know that declines the memory, the amyloid protein. So have you tried?
Sorry, I didn't understand.
The beta amyloid protein.
Yes.
Which decreases the memory. So have you done or are you planning to do any study where you give testosterone to the animal which already have this beta amyloid protein and see whether it improves their cognitive function, their memory?
That would be great to look at that. In fact, I mean, I have a longitudinal study that is different. It's just to look at sex differences, but as in aging. But as part of this, we're going to look at beta amyloid deposition, you know, as an endpoint, and see if, you know, there is a correlation between less amyloid and better cognitive function. But yeah, it would be great to see if you know, the testosterone treatment decreases amyloid deposition, which has been suggested by Christian Pike and other people. So yeah, it would be really interesting to see. Except that in our hands, testosterone didn't have any effect on cognitive function. So I don't know. Again, you know, you may have to give that for months.
I'm interested in the de novo steroid synthesis you mentioned in the brain. I remember seeing a talk last year from somebody from UMass who was doing bird.
Oh yes,
songbird
this is
brain.
yeah. So
And,
this is Luke Ramashili, and I collaborate with him. So he's
okay.
looking already at estrogen in the brain of our monkeys.
Do we know how it's regulated in the brain, and is the OVEX or GONEX model
We don't
really
know
a good model then?
We don't know if
Yeah.
if you vitamize a female, if there is a decrease in brain estrogen. That we don't know. Nobody knows.
Maybe it goes up.
Maybe it goes up. Yeah, I mean it's. So it's very interesting, and we don't know if it declines with age. Also, well, although there are some studies in humans. I think I remember that show a decline in brain estrogen with age, but you know, in monkeys there is no work for now that I'm aware of.
So that you measure temperature when you were talking about hot flushes.
Yeah. So
Where is the thermistor?
yeah, we use the telemeters that Jessica Mong is using. You know, so we record temperature for hours, like during the night.
Is it core body temperature or surface temperature?
It's core body temperature. Yeah, it's core body temperature. We have also, the monkeys are implanted with a chip that records skin temperature. So we have both measures, but obviously we need something, I think, like thermal imaging to actually see if they have hot flashes. It's hard to, you know, we try to trigger hot flashes by increasing the ambient temperature, and we see changes in core body temperature, but it's without the vasodilatation phenomena, it's hard to say it's a hot flash.
So how do you explain that estrogen has beneficial effects when it's used shortly after the decline estrogen levels, but if you wait a couple of years has no beneficial, actually has detrimental effects.
Well, this is what is, this is the assumption that everybody repeats, but actually in our hands, these over-optimized monkeys that were over-optimized for 12 years, they responded perfectly fine to estrogen for one task, not for the hippocampal task. So I think it depends on the brain region. It's not that, and we're not the only people. There was work from a Canadian team, and they were not looking at cognition, but they were looking at hypothalamic stuff in older monkeys also that had been over-optimized for years, and they were a clear response to estrogen. So I don't know. I'm not completely convinced by this critical window hypothesis. And in fact, the latest work in women, the Kronos study, I think, shows no beneficial effect of estrogen even if you give it at the time of menopause, if you give it to perimenopausal women. So, I mean, no negative effect, but no positive effect either.
Then why didn't we take estrogen?
Well, I don't know anymore. So for cognitive function, it's just we have not found the perfect regimen yet. Because in rodents, I mean, it clearly works like a miracle. And in monkeys, there are some. I mean, especially the RAP studies is really. I mean, they have dramatic effects of estrogen treatment on cognition, and they claim it's due to the cyclical regimen that they use, which is also possible. That instead of giving a steady dose of estrogen, you need to give a large dose first, and then that decrease. I mean, to mimic the cycle, the menstrual cycle.
Did you treat male monkeys with estrogen?
No.
No.
So the presumption is, I think, that the responsiveness changes over time because of receptor loss. But I'm not, I think a lot of that's anecdotal. I think a lot of that evidence is anecdotal. I don't think it's...
Yeah.
It's really not solid.
Yeah, it's not
Not
solid.
solid.
That's it, exactly. It's not solid.
And
Plus the idea, I'm really a strong believer in the pulsatile hypothesis because I think that's nature.
Yeah.
Constant level is not physiology.
But do we know that the receptors are gone after a long term of activity?
Yeah, that's what I'm saying. We don't really. I don't think there's good solid evidence that functional receptors are gone or that the receptors that are present are functional.
And they didn't really look at the receptors in old individuals, so we don't really know. I mean, I think they made it up as a way to explain what they think is.
Yeah.
But in rats, it would be easy to do in rats.
Yeah.
So,
But so I can't vouch for age other than about 3 months. But those earlier studies we did with estrogen replacement, those rats were ovexed and then went for, let me think. Well, at least 2 or 3 months before we got replaced.
Yeah.
So yeah,
That's
probably.
not long enough.
Yeah, that's probably not long enough.
Yeah.
But it didn't matter.
and let's not forget that for the WHI study, there were effects of treatment. They were negative, but
But it wasn't
obviously these
estrogen.
women were responsive to the treatment.
But it wasn't estrogen,
Well,
and
yeah,
it wasn't progesterone, so
well, but
you know,
even
it's-
with the, yeah,
It's a mess.
it's a mess.
It's a mess.
I know. Well, we can't make a lot out of that
Right.
study, but unfortunately.
I think those of us who unfortunately have gone through menopause, it makes a huge difference. Estrogen. All of a sudden you can sleep, all of a sudden you can think more clearly, which you can't separate the two very easily, but it's subtle as a train wreck.
Well put, Dr.
That's very good, David. Thank you.
Well, thank
Oh,
you.
There's some more? You have another question? Did you have another question? Okay, thank you.
Thank you.